All right, guys, it's your boy Tess back at it again with the DNA replication. Okay, so we got topoisomerase right here. Okay, what it does is it kind of unwinds the DNA strands a little bit, makes it a little bit looser up in there. So helicase right here can go ahead, split the brick. Now you got two strands of DNA. So you have DNA polymerase 3 right here. DNA polymerase 3 starts on the 3' end and it copies DNA. So it's just one continuous strand of DNA going down. That's on the leading strand. On the lagging strand, it's a little more complicated than that. So at first you have your primase right here, and it puts down a RNA primer. RNA primer allows for the DNA polymerase 3 to bind to it, so then it can begin laying down DNA. And then after it lays down the DNA, you have DNA polymerase 1, which pulls up the RNA primer that was placed down by primase. And then after that, you have a DNA ligase, which puts together the two fragments of DNA. The fragments created in the lagging strand are called Okazaki fragments.
RNA editing, it comes in 3 steps. The first step is initiation, and that is signified by the TATA box. And then the RNA polymerase binds to the TATA box with the transcription factors, and that comes across in the 5 to 3' end and creates a bubble where then elongation takes place, and RNA comes and is read off of this by the RNA polymerase, and it comes off and elongation, and then at a certain point termination happens, which is the last step of RNA transcription, and this then is sent to RNA editing.
So after transcription, the 5' prime receives a 5' cap, which consists of a modified guanine nucleotide, and then the 3' end receives a poly A tail, which is a series of 50 to 250 adenine nucleotides. So SNRPs, or small nuclear ribonucleoproteins, make up spliceosomes, which is this entire thing. The SNRPs are the circles, and what the spliceosome does is it bends the RNA into a loop, and then it cuts out the introns, which are the parts of the RNA that aren't encoded, and then it joins together the exons, which are the parts that will be eventually coded into amino acid sequences. So the end result would look something like this. And then this part is the exon, is the intron that's going to be cut out. So basically the spliceosome recognizes where to cut by recognizing specific nucleotide sequences, and alternative splicing, which is the insertion or deletion of certain exons, allows for the creation of different polypeptides, which leads to more complex organisms.
Okay, so DNA translation. All right, so we have our ribosome right here, and then we have our RNA strand, which gets fed into the ribosome. This is a piece of mRNA or messenger RNA. So the mRNA has sequences of codons, which are each 3 nucleotides long. So here's a codon right here, here's a codon right here. Each of these codons codes for a specific protein, well, no, not protein, amino acid, which is carried in by our tRNA or transfer RNA, which you can see right here. So the tRNA has an anticodon on it right here, which is anti to the codon, which is here. So it goes into the, let me get this in here right here, goes into the ribosome, pairs with the codon, and then drops off our little amino acid right here. So tRNA out of here, go on to pick up another another amino acid. So our amino acid is here. Our mRNA strand moves down to the next codon, which signals for the next tRNA to come in with a different amino acid on it. tRNA binds with the amino acid and then leaves the amino acid, which chains on like that, and then so on and so forth until the mRNA strand is finished, in which case it'll dissociate, and then our new protein will dissociate and go do its protein business.
For gene expression, there are different segments of DNA required for enzyme production. The TRP operon is responsible for producing tryptophan, an important amino acid. When more tryptophan needs to be created, RNA polymerase binds to the promoter and begins to transcribe the TRP genes into mRNA, which is then transcribed to produce tryptophan. When there are already enough tryptophan in the environment, it is now used as a co-repressor, which binds to the repressor, stopping the function of RNA polymerase. For a different kind of amino acid, lactose, a repressor is attached to the operator in the absence of allolactose, which is another form of lactose. When lactose or allolactose is in the environment, it binds to the repressor, causing it to separate from the operator, allowing the RNA polymerase to transcribe the lac genes, eventually producing lactose enzymes. Also, when there are low glucose levels in the environment, CAMP binds to the cap sites, speeding up the process for the transcription of lactose enzymes.